is composed of a series of distinct dynamic events that occur in sequential order, which allow the cell to effectively divide. In eukaryotic cells, this process is controlled by the mitotic spindle, a complex molecular device made up of microtubules, chromosomes, motor proteins, and two control centers called the spindle pole bodies. The two microtubule organization centers, the spindle pole bodies, are the mitotic spindle's most important tools for division. Throughout the cell cycle, these cylindrical structures regulate the processes involved in division while remaining embedded in the nuclear membrane. Microtubules span the spindle pole bodies, connecting to sister chromatids at the spindle midzone. Microtubules are dynamic polymers composed of the protein tubulin. The two ends of a microtubule filament are dynamically distinct. One end is highly dynamic and is called the plus end, while the other end is more stable and is called the minus end. An important property of microtubules is that they alternate between persistent phases of growth and shortening, a process known as dynamic instability. When coupled to a rigid structure, microtubules have the ability to exert forces by rectifying thermal fluctuations. Microtubule dynamics can be affected by intercellular factors. Various microtubule-associated proteins, plus tip-interacting proteins, and motor proteins can act to stabilize or destabilize microtubules. Thus, the cell cycle control system can directly regulate the key structural components of the mitotic spindle. In the initial stage of mitosis, termed prophase, replicated chromosomes condense. Next, during metaphase, microtubules arrange the cell's sister chromatids, which carry the cell's DNA on the spindle. DNA is a mechanical element that also plays an active role in spindle structure and function. The DNA is compacted into chromosomes, and the sister chromatids are bound together. They are only allowed to separate when all sister chromatids are correctly attached to the spindle. There are several motor proteins on the spindle that work in parallel to perform an assortment of specialized tasks inside the cell. They do this by converting the chemical energy from ATP into mechanical work. In particular, KIP1 and SYN8 are two motors that work to link microtubules together and guide them past each other. These plus-N directed motors are localized to the mitotic spindle and are predominant motors responsible for anaphase spindle elongation. Dynein is a -N directed motor which acts on the spindle via cytoplasmic microtubules. It is thought to be anchored at the cell cortex as it pulls on the cytoplasmic microtubules to help position the spindle and thereby contribute to spindle elongation. We are able to classify each phase of the cell's life cycle based on the length of the spindle. Cells in metaphase generally maintain a near constant spindle length that fluctuates between 1 and 2 microns. There are two stages of growth that the cell undergoes during anaphase, the phase in which the length of the spindle dramatically increases to 5 or 6 microns as the poles get pushed apart by the mitotic motors. SYN8 is known to play a major role in the initial stage of anaphase as the spindle's length rapidly increases and the small fluctuations seen in metaphase begin to disappear. KIP1 is predominantly active in the succeeding stage of anaphase when the growth of the spindle slows down until the spindle pole bodies eventually become two separate cells. We use fluorescent confocal microscopy to observe the three-dimensional processes that take place on the mitotic spindle. The two spindle pole bodies are labeled with fluorescent markers so that they can be seen when excited by a laser. A finding and tracking algorithm was adapted to extract the three-dimensional dynamics of the data collected. After tracking the motion of both spindle pole bodies, we analyzed the distance between the poles, the length of the mitotic spindle, as the cell divides. We see remarkable similarities in the elongation rate of the mitotic spindle during the onset of anaphase, despite each cell's many differences.